So it's my pleasure to introduce a relatively new friend, kind of, Dr. Steve Vollmer. Steve is the director of the School of Environmental, Coastal, and Ocean Sustainability, also known as ECOS, at Florida Atlantic University. Steve is an evolutionary and ecological genomist specializing in reef corals and their adaptations and resistance to disease, infection, and thermal bleaching. And he has been at Florida Atlantic since actually just last year. He received his bachelor's from Colorado State in 94, his master's from Cal State in 99, and his Ph.D. from Harvard in 2004. He did a postdoc fellowship at the Smithsonian Tropical Research Institute in Panama, and then spent the last 18 years at Northeastern as a professor of marine and environmental sciences and director of their bioinformatics program and associate director of the Marine Science Center. He is also a National Academy of Science Kavli Fellow. And I did want to mention two quick things. One was as I was kind of looking into Steve a little bit more, I mentioned this. There was a piece about him being a lifeguard in California, trying to figure out what he was going to do with his life. He had apparently sent in applications for grad school in Hawaii and where was the other? In California to do marine biology work, and heard about a program called 3Cs at Northeastern, which is a kind of intensive experiential research kind of focused program, which got him kind of getting into more of what he ended up doing, marine biology, coral biology, et cetera. And I did want to also mention one of his latest papers he co-authored with his son, who is a computer science nerd. I did want to go back to the surfer part because Steve was a surfer, and that kind of gives me a better understanding of why Steve has this kind of cool nerd vibe, which is great. And some of that may come across. So I could say a lot more about Steve. We're really excited that he's in Florida, and we are planning to do a lot of work, hopefully collaboratively, looking at some of the specific kind of more applied veterinary disease issues. Steve is with me and Josh Patterson, one of the scientific advisors for the Reef Renewal USA group. So without any further ado, I'll let Steve take over and introduce his talk himself.
Thanks, Roy. Yeah, I spent many years as Baywatch in Huntington Beach as a lifeguard, and connected me to my ocean, liked to surf, and then I figured out how to become a scientist and keep the adventure going. So I'm gonna share here. Let's see. You guys can see that?
Yep.
Perfect. I'm probably not going to be good at monitoring chat, but if you guys want to try to chat with me, Roy, if you see anything, we can stop.
Will do.
So thank you, Roy, for the introduction. It's nice to see everybody. And what I'm going to talk about today is basically the bulk of what I'm super interested in, which is coral disease resistance using the white band disease, which is in the center part of the picture here. It's a staghorn coral with white band disease, and how it's caused transitions from shallow coral dominated, elkhorn, staghorn dominated reefs to basically huge losses in the shallows. And I'll link it to thermal tolerance and what's happening in the Caribbean as well. And I'm specifically going to talk about our genomics work, identifying disease resistance genes. And then I'm going to make a pivot and talk a little more about probably what Roy wants to hear about, which is how we're using amplicon sequencing of bacterial communities, a little bit of AI as well, to identify rare pathogens in very complex coral disease communities. So I'll take us on a little journey today. Whoops, let's see. And so the basic outline of the talk is first to describe the system. So white band disease on staghorn corals will be the primary host pathogen system I'm going to talk about today. I'm going to present some of our work that we published in Science in the last few years on the genomics of coral disease resistance. So definitely a genomics-based paper. And then I'm going to pivot to talk about the white band disease pathogen work and how we're using machine learning models to identify coral pathogens from these complex communities. And then at the end, I'm going to show you a very recent paper where basically I'll show you that our machine learning predictions were actually right. And in a separate study, we basically got an association that machine learning told us it would be the pathogen, and it looks like it's behaving well. Sorry, it's moving around. So any discussion of corals, in my opinion, needs to start with describing the complex system that is the coral holobiont, and it helps us frame the kind of the problems that are facing corals as well. So corals, as many of you probably know, are an animal. The body plan is similar to an anemone, colonial, secreting a calcium carbonate skeleton. Inside the coral is the plant, more specifically a dinoflagellate in the family Cymbididiaceae. That's providing the coral with its nutrients, up to 90% of its sugar and carbohydrates. Corals also have complex communities of bacteria on top of them, inside their skeletons, fungi associated with their skeletons as well. So basically, they're a little ecosystem unto themselves. And if you think about the coral as a holobiont, it also helps you understand what happens when corals are breaking down or their relationships are breaking down, and it describes the two major threats to corals. So the first major threat to coral is the breakdown of the coral algal symbioses due to thermal stress, which causes a whitening of the coral that we call thermal bleaching. It's basically the corals digest or expel their algae and literally starve to death and ultimately die if it happens too long due to thermal stress. An equally important breakdown is the breakdown between the coral and its microbes. When I'm talking about microbes, I'm typically talking about bacteria in this talk. And breakdowns between the corals and their microbes cause disease symptoms and syndromes that cause pathogen infection and then opportunistic pathogens to come in and cause disease. And since we're talking to a Florida audience, many of you know that we've seen dramatic declines in Florida's reefs as well as Caribbean reefs as a whole. Recent estimates put us at about a 48% loss of hard or reef building coral since the 1980s. And these are some pictures from Ilse Kufner and Jean Shin originally showing these kind of transitions that we've seen on the left. These are like 1971 beautiful Floridian reefs dominated by hard corals in the top. You've got staghorn and elkhorn coral and a snorkeler. In the bottom, you've got a thicket of staghorn coral and then a hard coral in the front. You start to see transitions into the 80s. The transition that you're mostly seeing now was due to the epidemic of white band disease that took out the shallow water staghorn and elkhorn corals. You can see the fragments there. But bleaching and other chronic stressors to corals have caused larger losses. So the far right is kind of what our reefs look like now, where we've lost most of our hard corals and we've seen replacement towards macroalgae and soft coral communities in this case. To me, the three biggest events that have happened in the Caribbean are the white band disease outbreaks starting in 1979, which killed the shallow water staghorn and elkhorn corals. And you see a couple thickets down at the bottom in those two pictures, which is showing you thickets. These are actually from Panama, actually modern day, or at least in the last 10 years, where reefs just used to be dominated by staghorn corals at shallow depths, elkhorn corals at the reef crest, and white band disease took them away. You're starting to see the green plot starting in 1985 is basically the loss of coral cover. So we had pretty good coral cover in the 30s as back as 1985, and you start to see declines. The second major event in terms of a disease was the outbreak of stony coral tissue loss disease in 2014, another catastrophic disease that causes infections in up to 25 of the common reef building corals, causes devastating losses to them as well, and mortality rates in the 90s for many of them. And then just this chronic repeated mass bleaching events that have caused coral mortality accumulating in the 2023 catastrophic bleaching event, where basically Florida and the rest of the Caribbean cooked over 91-degree temperatures. We calculate heat stress and degree heating weeks, 22 weeks of degree of temperatures well above the bleaching threshold. So many months of bleaching and basically huge coral loss, including the potential functional extinction of the two shallow water Caribbean acroporids, the staghorn and coral and elkhorn corals that I'm talking about today. So the losses have been dramatic, and it's caused the majority of us coral biologists to try to figure out if corals can adapt and to survive future climate scenarios. So when we're talking about adaptation, we're wondering if natural populations can survive these repeat bleaching events or these catastrophic disease events, or if they needed to be boosted artificially through selective breeding programs and outplanting efforts. When I think artificial, I'm not talking specifically about introducing corals from another part of the globe, which has come up recently, or genetically modifying corals, because I'm a geneticist and I don't love that idea. And when people are thinking about adaptation, a lot of the questions focus on whether the coral genome and the coral host itself has the potential to acclimate, whether it can shuffle its symbionts, algal symbionts to help acclimation as well, or whether there's the potential of probiotic bacteria, for example, that will help corals resist disease infection or give them some additional thermal tolerance. So the holobiont has multiple mechanisms for adaptation and acclimation in the system itself. So where I'm taking us today is talking about my favorite system, which is white band disease. I've given you a little of the summaries, but these outbreaks occurred in 1979, spread across the Caribbean throughout the 80s. And by the end of the 80s, we saw mortality rates of 95% or more of the staghorn and elkhorn corals, our shallow water dominant corals. They're now endangered on the endangered species list as threatened. And over the last 20-ish years, I and others have been trying to understand the rules of white band disease transmission and who the pathogen is. So we've shown that the disease is highly transmissible, caused by a bacterial pathogen that can be treated by antibiotics. And you'll see today a large focus of my lab has been trying to identify whether the remnant populations have resistant individuals, and up to 15% of staghorn corals are highly resistant to disease infection by white band disease. Just to give you an idea of what happened, these are pictures from Gene Shin again in Florida. So in 1978, you see a massive boulder coral in the middle with a staghorn thicket behind it in the upper left-hand corner. 1979, white band disease is sweeping through that staghorn coral thicket. By 1988, the staghorn coral is basically all gone. The boulder coral's still there, but the skeletons are in place. And by 2001, you don't even see the remnant populations, even the skeletons left. And if you go across the Caribbean now and you dig your hands around reefs where you might expect to see staghorn coral, you can actually find the remnant skeletons of staghorn coral because they were the most dominant shallow water coral throughout the Caribbean. So a huge loss. Similar losses in the shallow to elkhorn corals. And just to give you an idea of the tempo of white band disease. So if you're close to the white band disease or the coral disease community, we like to call our corals by how the disease looks. So what you're looking at is a white band of dead and dying tissue. In some ways, coral biologists have basically split white band disease into two categories, white band disease type 1 and type 2, based on how the disease progresses. Part of the reason why I'm showing you this picture, which is an infection that's proceeding over the course of the night, is that literally with these types of infections, what you're seeing is a rapid tissue loss caused by the disease infection where the coral basically lost 5 centimeters of tissue in a day. It's not a discrete line. It's not a slowly advancing line. It's just basically loss due to infection. So if you think about a coral colony losing 5 centimeters a day at an infection point and coral growth proceeding at 10 centimeters a year, maybe a little bit more, you can see why the disease was so devastating and these infections are so devastating. The way this disease exists in populations, wild populations like we have in Panama, is as endemic infections. And in many cases, the coral will not completely die to white band disease anymore, the whole coral colony. You'll see losses along branches. You'll also see the coral actually resist part of the infection, and you'll see the disease arrest, and sometimes you'll actually see regrowth. So it's a very dynamic infection process in the best case scenarios where the coral actually is able to come back. So before I jump into more data, in terms of the transmissibility that I was talking about, the disease transmits via direct contact within the thickets. It can be spread in the water, but there's a key piece that the coral has to have injury or be lesioned, and we actually do this experimentally with airbrushes and water picks. And there's a variety of vectors for the disease, including corallivorous snails called Corallophilia abbreviata that can actually serve as a reservoir for the disease as well. You can starve a snail for 3 weeks after feeding it with white band disease, and it can still infect colonies when it is allowed to feed on a coral again. The disease is bacterial. We know that due to a variety of experiments, including using antibiotics to stop transmission as well as progression. In our lab, I'm not talking about it today, but we've manipulated quorum sensing molecules to turn on and turn off the disease. So stimulate quorum sensing can cause disease, turn off quorum sensing or inhibit it, and you can arrest disease transmission. But a focus of the talk today is going to be two things. One is how do we basically identify bacterial pathogens in complex coral microbiomes where the variety of the coral studies identify hundreds of disease-associated pathogens with most coral diseases, including white band disease. So this is what the state of the knowledge was, and I'll show you some advances on that today. And because we have so many disease associations, we haven't fulfilled Henley's-Cox postulate. We haven't cultivated a specific pathogen. We know it's bacterial, but we don't know who the specific pathogen is. And then as well, we know that staghorn corals can be highly resistant to disease. And if you're looking for some references, I left them at the bottom. So the first bit of data that I want to talk about today is the work we've done using whole genome sequencing and genomic approaches to identify the genetic basis of coral disease resistance using staghorn corals and white band disease. This was published in Science a couple of years ago. And I'll walk you through the steps. Basically, we compared two different populations of staghorn corals. Most of my work had been in Bocas del Toro in Panama, so we sampled 50 coral genotypes from 4 different populations in the region of Bocas del Toro to survey transmission experiments and to estimate disease resistance. We did a second experiment in Florida using nursery-raised coral genotypes at Coral Restoration Foundation in Florida. So 2 different transmission experiments to assay disease resistance and do this work. So what I'm going to show you is first how we basically assay disease resistance to get a score, and then I'll move us into the genomic approaches second. So if you're going to assay disease resistance in corals, the nice thing is that they can fragment much like a plant, so you can do replicate attempts for transmission. So we're doing these in tank-based transmission experiments. We did the 2 different experiments because we can't be in Florida and Panama at the same time, and we don't move corals. So we first did this in Florida, and then 2 months later, we did it in Panama, similar temperature profiles. In each experiment, we took 50 staghorn coral genotypes, 10 fragments per genotype, and we put them in experimental tanks. 5 of the fragments from each genotype went into healthy dose corals, so our controls. 5 of the fragments went into tanks that were going to receive a disease dose. I'll show you what that looks like in a second. And what we were basically measuring as metrics of disease resistance was censusing the rate of disease progression by monitoring it over days. We were sampling the communities, picking polyps and looking at RNA and bacterial changes in the tanks as we went. I'll show you some data on that at the end. We pulled disease fragments out of the tanks as they became infected, so we don't amplify the disease in the tanks. And I'll show you the statistics, but effectively, disease resistance is scored based on the number of fragments that became affected out of the 5 that went into the tanks. So in conducting the transmission assays, you rack the corals, color code them by genotypes. You have to water pick them to cause an experimental lesion, which is a requirement for transmission. You put the healthy corals in the recirculating tanks. You see this was at the Florida Keys Marine Lab in Florida. You have to have strong replication, so lots of corals being killed for this experiment, especially in Florida. These were coming from nursery-raised fragments, so we weren't causing any harm to natural populations. The experiments themselves, each experiment was 500 fragments, which is 50 genotypes, 10 per thing, spread across 10 replicate tanks. Quite a undertaking. The way we make disease doses, which will come back a little bit later when I talk about how we are profiling disease, is we take healthy corals from the field and create a mixture by water picking with sterile water, their tissue to make a healthy slurry. We take 20 diseased interfaces, and we create a disease slurry. We call this the disease slurry of pestilence. We normalize the amount of ocular density so the doses are about the same. We dose each of the tanks with a similar dose of tissue and pathogen or bacterial communities, and then we look at how the transmission proceeds. So the disease appears after about 2 or 3 days. And basically, all the time we're recording, sampling RNA and DNA and profiling the immune response and IDing bacterial pathogens. So this is what disease transmission in the tank looks like. This is me being silly and my graduate student Brescia behind me. She's not surprised. This is 3 days into a transmission experiment. She's exceptionally tired because we've been picking polyps off of all 500 corals for the last 2 or 3 days. So not excited, just very tired. And all that work gave us this figure. So what I'm showing you here is the y-axis is fragment survival. The x-axis is experimental day. The two experiments, the Florida's in blue, in Panama is in orange. And basically, this is the infection rates. Each one of those lines is one of the individual genotypes out of the 50 genotypes for each species. And you can see that the Panama experiment proceeded more rapidly than the Florida experiment in this case, with the medians in the middle. What this allowed us to do is to basically get a normalized disease resistance score across the two experiments. When you do this and do the survival modeling, the hazard modeling, you basically get that 6% of the variation is basically coming down to genotype. There's a lot of experimental variation across these two experiments because they're at different locations with different pools of disease and different genotypes. So 28.8% was experimental. But what's going into our disease-resistant assays is panel B, where we basically take normalized disease-resistant scores, and we've taken this as an inverse of infection. So in Florida, the individuals that are higher are more resistant. The individuals at the bottom of the graph are more susceptible, and same for Panama. So it's those disease-resistant scores that are going into our genomic analysis, trying to identify genes associated with disease resistance. We move into a genome-wide association analysis to basically identify the diseases or the genes associated with disease resistance. Sorry. If any of you work in non-model systems and are doing genomics, having a reference genome is actually great. We did not have one, so for this study, we had to basically de novo assemble our own staghorn coral genome. We did this using a nanopore-based approach. We assembled a reasonable staghorn coral genome. It's 308 megabases, so corals have relatively small genomes. We identified 28,000 or so genes. And if you're interested in genome assembly, nanopore is pretty powerful. We've been able to do this as well. We now have a more chromosomal scale genome than when we first published this one for the staghorn coral. The next thing we did is we basically used whole genome sequencing to profile all 48 corals from Florida and 48 corals from Panama. We subsetted 2 out for each species so we could fit them into a 96 well run. And we didn't want to make any assumptions about having low coverage, and we didn't want to do any approaches like RAD sequencing. We wanted the highest quality whole genome sequencing data that we could get, and we could afford it in part because the genome was small. So the average coverage that we had across our samples was 75x coverage, and the majority of individuals had 20x or higher coverage across their genome. So it's a pretty solid data set. With our SNP calling, we identified 1.2 million SNPs at allele frequency of 0.5 or above, or 0.05 or above. Gave us a lot of variation to look at. Unlinked SNPs, 54,000 SNPs in the genome. The bottom plot there is interesting because it shows that there's strong genetic structure between Florida and Panama. And if you're doing any kind of genetic association studies, you need to account for population structure before you go in, which we did in our models. The difference between Florida and Panama is in a global FST of about 0.15. So they're pretty genetically differentiated. In the Caribbean, actually, staghorn corals probably fall into at least three distinct genetic populations. And the data I'm going to show you today is interesting because after you can control for the genetic structure between populations, these are shared disease resistance genes that are shared between Florida and Panama. There might be unique variation within, and that's something that we're within populations that we're exploring now. Our genome-wide association studies were basically identify SNPs associated with the disease resistance scores coming out of the transmission experiments. We use latent factor mixed models and basically identified those genes. So one of the reasons why our results were highly publishable and surprising is people had tried to do similar approaches in other coral species, mostly focused on thermal tolerance. They had applied relatively robust gene sequencing techniques, relatively deep data sets, and in those thermal tolerance profiles, they didn't identify any genes associated with thermal tolerance. Whereas when we looked at disease resistance in this system, we saw at least 37 highly significant SNPs associated with disease resistance. Those SNPs fell into 10 linkage groups or 10 chunks of the genome spread across 7 chromosomes, and there were 8 genes in those linkage groups that contained these SNPs that are associated with disease resistance. It's a very complicated plot. I'll summarize it a little more, but basically the panel A is showing you the significant values. Each one of those gene names is basically in red is one of the gene names. I'll decipher that a little bit. The bottom plot is effect size. So any of the red dots above with a positive effect size means that the variants are associated with the rare variant. And if they're a negative effect size, those are SNPs associated with the common allele or common variant. So in a nutshell, we have all these SNPs. We have 10 linkage groups. And when we started to look at what genes had variation associated with disease resistance in the protein coding genes, we identified 4 genes associated with resistance that also had a function involved in immunity. So the gene names are listed, but the functions are more interesting. So PTPRD is a tumor suppressor and immune regulator. SEG is a immune regulator and regulates phagocytosis, so the digestion of potentially invading bacteria. AP3D1 is involved in lysosomal trafficking and endocytosis, so again, a digesting process. And then LRP2 is something that will activate complement and NF-kappa B, so 2 common innate immune pathways. So we're seeing disease-resistant variants associated with disease resistance in these genes, but we also had RNA sequencing data so we could ask if those genes were differentially expressed due to disease transmission. And the surprising piece is that none of these genes were differentially expressed. So they seem to be constitutively expressed, and what we think is happening is that it's variation in those protein coding genes that's driving resistance, not the differential expression of those genes as well. And I think probably the last piece of this study that I think is really interesting is that we then went back and we genetically modeled how well our disease resistant associations can predict disease resistance. So the polygenic score is basically the effect size score of all of those disease resistance genes, how they're adding up in an individual coral genotype. And then the y-axis is basically the observed disease resistance with a little bit of fancy genetics thrown in. And what it's showing us is that our top 10 SNPs, those top 10 chromosomal regions and the top 10 SNPs with the highest effect sizes, can actually predict the disease-resistant outcomes in the tanks with 76% accuracy. If we extend that to be other SNPs of moderate effect and include it to be 63 SNPs, we can actually predict 85% of the observed disease resistance. In terms of the data, what that meant overall is that out of the 96 corals that we screened, 9 of the ones from Florida, so 9 out of the 48 were highly resistant, 5 from Panama out of the 48 were highly resistant. Those are the ones that people will want to use in outplanting efforts, whereas 7 from Florida, and 8 from Panama were highly susceptible. But this is giving us a genetic tool to basically screen corals without having to do those complex transmission experiments. So before I pivot to the pathogen work, the major findings here is that disease resistance is genetic and heritable, and has links to immunity, and that we can now identify disease-resistant corals based on as few as 10 genetic loci. And a huge part of the lab now is trying to understand whether there's any fitness trade-offs. Are more disease-resistant individuals have lower rates of growth? Is there any link or interaction with thermal tolerance as well? We think they're independent. We have data that suggests that disease resistance and thermal tolerance is independent, but give us another year or so, and we can look at that as well. So now I'm doing my hard pivot, and I'm going to talk about the work we've done trying to find who the white band disease pathogen is. And coral biologists have been strongly hunting for coral pathogens in these complex communities. And I would say that we've been pretty unsuccessful at identifying keystone coral pathogens in traditional frameworks like fulfilling Henley's Cox postulate, where they have to be cultivated and reinfected. So this isn't just a white band disease problem. This is a stony coral tissue loss disease problem as well. And it's not despite a lack of effort. So in my labs and others, the results basically look like this. Early 16S culture and transmission data, we're pointing to a Vibrio, first called Vibrio schloei. It's probably actually Vibrio coralliticus, an opportunistic pathogen on many corals. But then people like my lab and others started doing more modern RNA sequencing with amplicon sequencing. And every time we would look and compare disease corals versus healthy corals in our system, we'd identify hundreds of associations. And so I'm giving you the list, but Vibrios, Flavobacters, Campylobacters, Francisellus, Pastorellis, if you're dealing with marine pathogens, you're seeing a lot of familiar families, um, which is fine, but you're not really getting what we would could be a keystone pathogen. And kind of the way I think about it is when we're actually sampling disease corals in the field, or even in our transmission experiment, we're seeing shift in bacterial communities that are basically progressing over time. So on the left with the homeostasis, that's a healthy coral dominated mostly by mutualus. You then have a coral individual becoming infected. And in a perfect world, you get a high abundance keystone pathogen, which we kind of expect for many diseases, but doesn't always happen as the case. But as the keystone pathogen infects, it starts to recruit other things like opportunistic pathogens, potentially due to quorum sensing, and then you start getting these complex mixtures. And you probably can't see my pointer, but if you can, if you took a slice right down the middle where the keystone pathogen is there and the opportunistic pathogen, and that's where you were sampling and profiling those bacterial communities, you're going to get these complex mixtures. And I think this is really what's happening when we're actually sampling disease. So we need a tool to basically sort out who a keystone pathogen is from these complex associations. So after many years of just trying to look at disease associations the old way, which is through differential abundance, we started incorporating machine learning models and deep sampling to try to basically improve what we were doing. So this is a recent publication. As Roy said, that's my son there, Miles Vollmer. First author is my postdoc, Jason Selwa, and 2 students and myself. And what we had accumulated for these machine learning models is probably the deepest sequencing of a disease to date. There's some great Skittle-D sequencing as well. But basically, we knew we had to have numbers. So this is all data from Panama. And we basically went to the field and sampled healthy and diseased corals from 4 different sites at 4 different time points over 2 years, spread out about every 6 months. So January, July, January, July. We sampled 20 healthy corals and 10 diseased corals from each one of these sites because we wanted to see if there were asymptomatic healthy individuals that looked like they had disease in terms of their microbial profiles. If you sum everything up, we're talking about 400 samples. So 143 diseased, 269 healthy corals. So it's a pretty healthy data set. It's all 16S amplicon sequencing of the V3, V4 region. The depth is pretty good. And we used a mixture of typical disease association work, like looking at differential abundances, but then more importantly, we tried something new, which is ensemble-based machine learning models. So without going into it too deeply, but I'm happy to talk about it. If anybody wants to geek out on this, I think it's kind of interesting. What we did is we basically took two or three inputs. We knew the corals were either healthy or diseased, so we were scoring them as healthy and disease. And then what we were doing is looking at how well the predictions was using only um, the abundance of the bacterial types. So imagine taking a matrix, a count matrix of all of the bacteria associated with all of these 400 some odd samples, just the bacterial communities, and seeing which parts of the bacterial communities predict disease outcome, healthy or diseased. We applied 6 common or 7 common machine learning models. We were basically looking for accuracy predictions to see how well we could predict disease outcomes purely based on their bacterial communities. That's part 2 here. We then used a tank-based transmission experiment to see if our models were predicting something. That's kind of a sanity test. And then I'll show you work that shows you kind of like whether it vibes with what you would expect of a pathogen. We also had some really interesting data just to quickly talk about on how disease prevalence is changing in these wild communities in Panama with these endemic infections. You see in panel, well, we had some heating events. So these are degree heating events. So higher number of degree heating events is more time spent above the threshold. So we had some variation. These are the actual white band disease prevalence values from the field. So you see them going up here in January, down, and then peaking again. But there was a significant correlation between the degree heating weeks spent above 30 and how much white band disease prevalence is in the field. So there's a thermal link to this as well, either due to coral stress or maybe advantaging more opportunistic pathogens. But here's the machine learning work. So to explain this, basically, these are the different machine learning models that we compared. The null is basically our null expectation. This is a typical train test kind of splits with lots of replication and also tuning. These are common machine learning algorithms. This is random forest. This is a lasso regression. Basically, this is nearest neighbors, just some common things. And what a lot of machine learning practitioners, especially on the computer science side or on the health informatics side, what they're always interested in it is accuracy, right? How well does some feature set that I load in, where our feature set is actually the bacterial abundances, how well does that predict some outcome? And the good news is that our bacterial communities actually can predict disease or healthy state with high accuracy. So overall, the bacterial communities are predicting whether a coral was diseased or healthy with 97% accuracy. So the machine learning side is working, right? The other thing, which I'm not showing you, is that there was equivalent accuracy between the healthy populations and the disease populations, which for us was a surprise because we expected some of those healthy individuals to actually be asymptomatic and we expected them to type as disease, and it wasn't happening. So what that means is that the bacterial communities are actually predicting disease outcomes, and it looks like if there's a transition from asymptomatic to disease, it happens very quick. The other secret sauce in these machine learning models is we basically can create ensemble models by testing for equivalence. And so in our case, these top 4 models were equivalent with each other. And we can basically ensemble these models and then basically look at their performance to try to predict who the most important bacteria are. So that's something that we've been piloting, where most machine learning practitioners are trying to get accuracy of the models. We're actually doing feature set exploration. We want to know from the data who the most important pathogens are. So in a coral community, you've got the pathogens and their abundances as features. You've got hundreds of features that you're basically putting into these machine learning models. And then you basically want to identify who the top ranked features, or in this case, bacterial ASVs are. And a lot of times people use only one model. So like they'll, use random forest, for example, and they'll say this is the top pathogen. What we do by ensembling is we're able to get kind of a total evidence idea. And that's what you're seeing here in A. In the equivalent models, we're basically looking at the ranks where you want to be a higher rank. And so Cystinophilum literale was the top rank across all the models. And then as it goes down, they get worst ranked. Okay. So Cystinophilum literale is our top ranked disease-associated predictor. Vibrio was second. In the second panel here, B, these are shapely values. So these are machine learning-based predictive values called shapely values that we use pretty extensively. The nice thing about them is you can compare them across multiple models. And so the way you interpret these is as you go further up and to the right, that's a more important predictor of disease. So in that individual sample. So each one of those dots is an individual sample. So according to the model in panel A and B, those are the most important ranked pathogens from top to bottom. Then we threw in our sanity checks. So panel C was whether or not we saw strong differential abundance differences between diseased and healthy corals. So those are log-fold changes from the field in those samples across all of our time points. So each time point is a dot. And Cystinophilum literale passed, Vibrio passed, and basically the red dots are things that were significantly different at each time point in the field. And then the last sanity check that we did is we had independent tank transmission data that was not used in the machine learning models. And we asked two questions. Were they differentially abundant due to disease exposure versus the healthy control tanks? And were they significantly associated with disease outcome? And there were only three of the predicted ML top pathogens that passed that test. Our two top ranks, Cystinophilum literale and Vibrio, and then this Neptunobacter that I'm going to ask you to just forget about because it's a weird nuisance. But these two we think are remarkably important. So this is what was happening in the tanks. You look at the tank data, here's what was happening in the field for Cystinophilum, and you see it's at much higher abundances than on disease corals than on healthy corals in blue. And basically that number is basically a zero because it's a log to counts per million value, and that's basically zero. And this is the tank portion of this. When the corals came into the tank, we sampled them. This is the healthy control exposure. These are corals in the tank in the whatever the dots are there that didn't get disease, and then the red dots in the tank up higher, those are the ones that got the disease. So for Cystinophilum, you can see the ones that had disease had high abundances of Cystinophilum. Now we're going to say that the secondary opportunist is going to be a Vibrio species, this ASV8, because you can see the Vibrio abundances even on healthy corals were higher. They were still differentially abundant in the field. They were at higher densities on diseased corals in the field. And in the tanks, they were at higher abundances as well. And they were still disease associated. So when we published this paper, we said, we think that the pathogen is a Cystinophilum, and we think the Vibrio is a secondary opportunist because that's what Vibrios typically do. But we had to leave it there. So the last piece of evidence I'm going to leave you with is a very recent paper, used none of the fancy machine learning, but is an independent experiment going back to our Florida transmission experiment, where I'm going to show you that the Florida transmission experiment basically recovered this Cystinophilum as the pathogen. So it's the same model of the disease resistance data because it's from the same experiment. Healthy doses, disease doses. In this case, we sampled at day 0, day 3, and day 7. We did amplicon sequencing of those same fragments in that disease resistance assay. We can look at differences due to disease outcomes. We could identify things as early responders or late responders. We can identify differences between disease outcomes within disease-exposed tanks. So these guys got disease, these guys got exposed to disease but didn't get the disease. And we could even go backwards and compare whether or not the pathogen came in on the doses, comparing the abundance in the disease dose versus the healthy dose. So pretty complicated models, but pretty thorough as well. You see lots of shifts in the tank. I'm not going to belabor this too much, but here are the differences in the homogeneous doses of things ranked by genus and family. you see strong shifts over time. Um, and what I'm going to show you here is that sustenophyllum is a very weird color. It's this color here in the trichocales. It's that kind of the purple. And what you can see is in these raw abundance plots, it's very rare. Um, it wouldn't be catching your eye if you weren't looking for it. But you do see some things happening. So these are in the disease-exposed corals at the very end, and there's a lot of thalassodias in this brown that are strongly associated with disease outcomes. There's some vibrios in there as well, down here. And if anybody tracks the coral literature, people like rickettsias. They think rickettsias are really important, especially with white band disease. So rickettsias are basically these highly pathogenic, tank-loving bacteria, especially in staghorn corals. And basically, the rickettsias had no association with disease resistance, but they flared up in the tank at disease 3, and then they went away. Sorry. But this is the big result. So what we're showing you here is top rank pathogens, no machine learning, and whether it was associated with disease outcome, whether it was significantly different in the disease tank between disease and susceptible individuals, and whether or not it differed significantly on the incoming homogenate doses. And the only pathogen that passed this test was sustenophyllum, this guy up top. The ones in the next category that we call white band disease associated opportunists, they include lots of familiar players like those thalassodias. And see this Vibrio here, Vibrio species ASV 134. That was also strongly associated. So again, another Vibrio. And the interesting thing in these data, the thing that surprised us most is if you remember the machine learning model, sustenophyllum was the top pathogen, a Vibrio was the second. This sustenophyllum that came out of the, and that was in Panama, this sustenophyllum that came out of this Florida transmission experiment is basically the same ASV as the one we detected in Panama. It's got one base pair different over a 350 base pair fragment. And so that's the first example of an association over a basin scale disease in any coral literature that we know about. So basically, we're basically confident that sustenophyllum is the white band disease pathogen, a keystone pathogen. We also had a Vibrio in Panama, but this is not the same Vibrio as the one we found in Panama. It's actually very different. It's probably about 10 base pairs different. So it's just another associated Vibrio coming along for the ride with the infection. And what we're now doing is continuing these novel machine learning approaches to identify the associations, but we're moving to cultivation and single strain transmission. If anybody does coral cultivation work and microbial work, you know this is hard. This is our first attempt to basically cultivate sustenophyllum. These are individual streaks of strains, and none of these were sustenophyllum. So it's going to be a long hunt. But we feel like that's what we need to do to basically identify and finally fulfill sustenophyllum as Cox postulate level pathogen associated with white band disease. So I think what I've shown you today is that we can use genomics to basically start to look at genes and genes associated with disease resistance and an easy step to thermal tolerance and start looking at trade-offs between disease resistance and other functional fitness traits like growth and combined traits. We know how to basically grow corals at scale, especially in Florida. We're actually funding that at a reasonable rate. And it's a pretty logical step to start looking at phenotypic variation in thermal bleaching responses, like you're seeing in these brain corals and these boulder corals here from the 2023 bleaching event. And so we're moving our disease resistance work to try to apply the same genome-wide association studies to look at thermal tolerance and do it across a wide range of species. You can actually use the same approaches and develop these polygenic score techniques and actually look for combinations between disease resistance and thermal tolerance and use it in outplanting efforts. The other thing that's unique about Florida is that we know how to restore reefs at scale, but we aren't often using science, the best science to basically back up our restoration efforts. You're seeing on the right a large number of people outplanting corals In many cases, we're stuck at trying to maintain just genetic diversity in our outplanting efforts. And I think the disease resistance work and future thermal tolerance work argues for the need to build in resilience in terms of disease resistance and thermal tolerance as well. But given that most of you are scientists on the call, this is probably going to fall within our community. One of the biggest gaps that we have now, and especially in recent times, is that we're not matching our investment in science or outplanning to basically meet the scale of the problem. And so in Florida, they're doing a pretty good job. Florida-wide, they probably invested about $40 million per year in reef restoration efforts. And that's for a reef-based economy that produces $8.6 billion back into the Florida economy and ecosystem services, but also just tourism. That'd be a 0.5% of reinvestment, pretty poor if you were running a business. And I pulled some back of the envelope comparisons between what we're investing in our U.S. forests. So we spend about $9 billion a year to maintain our U.S. forests. It's about a 50 billion economy. So that's a reinvestment rate of 17%. So if we want to shift our corals back from to coral dominated reefs, we need to invest back into our reefs, into our science, and also into our outplanting efforts to try to match the scale of the problem. Thank you. Can I take any questions? It felt very COVID-y to give the talk like that and not see any responses.
That was awesome, Steve. Appreciate it. Let's see. We got Cheryl. Cheryl is online. Cheryl, you want to go ahead?
Hi, Steve. Nice to
Hi,
see you and
Cheryl.
nice to know you're in Florida. That was a great talk. The question I have, so you now have identified what you're classifying as disease resistant corals.
Yeah.
How, and so you plant a bunch of these. How do you account for pathogen evolution?
Yeah. I mean, I think that you probably could get co-adaptation. So anybody that's going to be highly resistant, the assumption is not that they're going to be highly resistant, but I think if you're talking about a single pathogen like cystenophyllum, that the assumption that you can make is that we're profiling resistance to what is we think is a cystenophyllum pathogen. I guess the nice thing about those disease transmission experiments as well is because it's not a single strain and you're giving them a cocktail, you're probably giving them a more divergent cocktail. But the other thing that heartens me a little bit is we're doing these experiments in 2022, which
Mhm.
is 30 years into the white band disease epidemic. And
Mhm.
so I guess the pathogen evolution piece is really interesting. But I think that we're probably looking at an endpoint or a very evolved point. And so we're actually probably surveying disease resistance after 30 years of pathogen evolution. I think the idea that's cool, it's not a cool for the corals, but
Right?
with Skittle D, where we had a more recent infection, we probably could have actually caught a more recent, and you know how, I mean, you're so actively involved in this too. The idea of like every coral that's sitting on the reef that has seen Skittle D is now resistant to it. I think that's not how we should be looking at that.
No.
It's variation in resistance. So I'm tiptoeing around your question a little bit. We have tried to play with some experiments to see if we can actually get more virulent infections by doing multiple
Mhm.
ranges of pathogen infection. There is I think what actually is going on in those types of experiments, which would be worth looking, is I think the pathogen evolution or the increased virulence is in the recruitment of opportunists. So
Okay.
I think that in
Mhm.
tanks you can get amplification, but it's a very cool idea.
And you can also go the other way too. It could become less virulent
Yeah,
and just sustain itself and degrade the fitness
yeah,
of the organism against other things as well.
yeah.
Can I have one more question or you need to go to somebody else?
No, go ahead, Cheryl. I think Mark, I see Mark's hands up, but I think he's
Um,
fine with you. Wait, go
and
ahead.
this is more of a general question
Yeah.
because we've been working with some proteomics of stony coral tissue loss disease. And we actually had the opportunity to have some very rich monitoring data before, during, and after at different sampling times over a year or more. But what we are seeing that much like I think of human medicine and maybe, I don't even know, veterinary medicine, maybe domestic ones, we're seeing that what those, if you treat them as patients, the colonies, their history impacts what we are beginning to see and helps disentangle some of what may actually be setting them up, or because we've seen things go into remission, things that were healthy become sick, and so forth. And I just wonder, with your huge data sets, many more pieces than we do you have you looked at the idea of looking at these colonies as patients with a case history or a patient history associated, and how that may affect your outcome?
Yeah, I mean, I think just two quick pieces of that because it's something I've thought about. I mean, I've always been struck. There was a paper by Mauricio Rodriguez Lanetti suggesting that there could be memory in infections in aptasia. So I think that's a component that people haven't tracked down. The kind of the data sets that we've seen with our restoration partners, they're tree-based like scores. And so it's almost like, do you treat the fragments as independent units? I will say that for a while, I was trying to monitor actual colonies in the field
Mm-hmm.
and for cervicornis. So this is in Panama, and it wasn't a strong. The problem with the data sets is, it's the probability of infection is
Yeah.
low. And then
Mm-hmm.
you see, so there wasn't ever a strong signature of a colony getting infected and then never getting infected again. It was kind of a
Mm-hmm.
haphazard thing when you looked at it. So I think it's only in the situations like you're talking about where you get these sublethal. It'd be interesting if you get these sublethal events and then you go back and assess it again. I think that's the
Mm-hmm.
only way to really track it, or unless.
Because we were doing repeated measures on the
Oh
same
yeah,
and these were faviolatas, so
yeah,
not exactly a cervicornis. But
yeah,
I found it, if they had bleached before, had an effect. Um, it was very telling that when we started treating them in that fashion, there was a little bit more clarity in our data.
yeah. I think that type of going back, repeated measures across the same genotype where you're looking at those sublethal responses, if you had enough data as you suggest, you can fish out those patterns, but as you know, they're just hard to
Yeah,
come by.
right. Thanks very much, and thanks for your
Yep.
talk.
Good to see
Great,
you.
great seeing you, Cheryl. Thanks. Hey, Mark, you have a question?
Yeah, thanks very much. Yeah, quick question. With all the interest in Endozoicomonas as, potentially one of the main probiotics, what are your thoughts on picking up 3 different Endozoicomonas ASVs as in your machine learning as being identified as potentially pathogenic, or, in the disease corals?
Um, there's something I didn't tell you in that talk, and as I was giving it, I think here's my thoughts as Endozoicomonas. That best data came from the Red Sea, right? And if you look at different coral species, you'll find that some of them are Endozoicomonas dominated. Cervicornis is one of them. If you look at other Caribbean species, they're not. What we actually did in that one study is we were trying to find probiotics. And what we did in that same experiment that I didn't describe is we actually asked, did any specific bacterial pathogen any bacterial ASV genus or family contribute to the disease outcomes in the tank? So we took T0 samples from those genotypes and then looked, and we got 0 probiotic associations. I probably should have done that. So I think I've been trying to think, much like I think what you're getting at is like, how would I find probiotics other than, cultivate something and then hope that it's a probiotic and show the experimental, like do it with a machine learning model. It's not coming out in our cervicornis data sets at all. There doesn't seem to be a strong signature. We have some evidence that there might be something along the same lines that you were looking at in terms of the Skittle D. But yeah, we should probably talk offline because you're probably looking at the machine learning model and seeing a positive association. That would be the one that you would expect as well. And I think endozoicomonas is actually, when you look at the cervicornis data, it's a hallmark of a healthy coral. And when corals become diseased, they get dominated. So it's almost like it's a biomarker for health.
Yeah.
But they don't seem to be helping. They just seem to get taken out during an infection or something else dominates. Did that help?
Yeah, that's perfect. Yeah, I'll ping you an email right after this.
Yeah.
Great talk.
Great. Anybody else have any questions? I don't see any questions in the chat or hands up. So I think you are off the hook, Steve. Appreciate it, man. Hopefully I'll get a chance to go over there, or you can come over here. We can chat.
Yeah,
I have a lot of follow-up questions, obviously, but I'll talk to you more later.
Yeah, let's talk offline. Thank you, everybody. Talk to you later.